Cell division by Pratyusha Manthena.
Vinayak Sahal.
Dheeraj Nuthakki.
Mitosis is a form of cell division, one of the two that we'll be talking about today, and the purpose of mitosis is to create two identical somatic cells. Now, somatic cells go through mitosis to create two daughter cells that have the exact same DNA, making them completely identical. In the end of mitosis, both cells will have a complete set of 46 chromosomes, making it a diploid cell. Mitosis is one part of the cell cycle. The cell cycle consists of mitosis and another part called interphase. Interphase consists of three different sections, G1, at the S phase, and G2. During G1, the cell grows and creates parts for cell division, and in the S or synthesis phase, the DNA replicates to create two copies, which are required for mitosis because we need two different copies for two identical somatic cells. And in the G2 phase, the cell continues to grow and make final preparations for mitosis. The first phase of mitosis is prophase, and during prophase, the centrosomes start to unravel their spindle fiber, which will be used later in the mitotic process, and the nucleolus disappears, and the chromosomes start to form. The chromatin, which refers to the giant mess of DNA in the nucleus, will start to bind and form a double chromosome. A double chromosome is made up of two sister chromatids. When attached, these sister chromatids will be called a chromatid, but when they're separated, it can be considered a chromosome. And the two sister chromatids are completely identical. They're just a copy of each other. During prometaphase, the next stage of mitosis, the nuclear envelope disappears and the centrosomes start to unravel their spindle fibers and start connecting to the centromeres of the chromosomes, which start to move towards the center or the equator of the cell. During metaphase, all the chromosomes are lined up at the equator of the cell, and the spindle fibers are attached to the centromeres, and they are lined up perfectly in line in the middle of the cell. During anaphase, the replicated chromosomes split apart and are pulled towards the poles of the cell by the spindle fibers, and the centromeres dissolve. During this process, cytokinesis also starts to occur. The last phase of mitosis is telophase, and the chromosomes are now at the two opposite sides of the cell, and through the process of cytokinesis, the cells move apart and form two different cells. During this process, the nuclear membrane continues to develop and form around the chromosomes, and the chromosomes unravel and become chromatin again. Now we have two identical cells that are diploid cells with the exact same DNA in both. Now we're going to talk about meiosis, which is another type of cell division. In meiosis, the end result is 4 different gametes that are produced, and each of them does not have the same DNA in them because of different events that occur to create genetic variation. The result is 4 different haploid cells, not diploid cells like mitosis. In haploid cells, there's only 23 chromosomes instead of 46. Meiosis is just like mitosis, but 2 times, because we want 4 different cells. So we have two times meiosis, so we go through prophase, metaphase, anaphase, and telophase twice rather than just one. The first step of meiosis is prophase 1. In prophase 1, by this time the DNA has already replicated, and we have the nuclear membrane which starts to dissolve, and the centrosomes start to release their spindle fibers just like normal prophase. During this process, something important happens that allows for genetic variation. The homologous chromosomes that start to attach to each other to create a tetrad, and this allows for crossing over of the genes. The alleles that are similar on both chromosomes cross over and change places and switch spots. This allows for genetic variation, and the result in the end means that the DNA in all 4 cells is different. During metaphase, the tetrads are lined up in the middle of the cell, and the spindle fibers are attached to the centromeres. The independent assortment of the chromosomes in the middle allow for genetic variation. There's a 50% chance that a particular gamete will receive one half of the homologous chromosomes either from the mother or the father. So this allows for genetic variation because we do not know which gamete these will end up in. Now, during anaphase 1, the spindle fibers pull on the homologous chromosomes, and the homologous chromosomes split apart. Then they move towards the poles of the cell. During telophase 1, the homologous chromosomes continue to separate and reach the poles of the cell, and each pole now has a haploid number of chromosomes. The cell starts to move apart from each other and create 2 different cells through the process of cytokinesis. Now we have 2 cells, and we can move on to the second part of meiosis. During prophase 2, the cell gets ready for the rest of the second part of meiosis. However, between the 2 stages of meiosis, the DNA doesn't replicate again, which is different from mitosis. During metaphase 2, the chromosomes line up at the middle of the cell, and the spindle fiber is attached to their centromeres. During anaphase 2, the chromosomes are pulled apart by the spindle fibers, and the centromeres dissolve. And this begins the process of cytokinesis as the cell starts to pull apart. The last part of meiosis is telophase 2, where we now have 4 different gametes, all which are haploid cells with only 23 chromosomes, and the DNA in each of the gametes is different, allowing for genetic variation of the cells.